Hey, this is Mr. Armand, and this is Section 4, Chapter 5 of PCR. Today we're going to talk about thermocyclers and what they are, what do we use them for, all kinds of other crazy things. Alright, so the way I like to think about thermocyclers is kind of like this, and this is kind of a strange analogy, but if you've ever been on a roller coaster ride and you it's like the most amazing roller coaster ride in your life. You know, you did all these twists and turns and ups and down. You went so fast, but by the time you got done and you were so exhilarated with it, you were so excited about the roller coaster ride, like you got to talk to your friends about it and just like they'll ask you, well, why is that roller coaster ride cool? And you're just like, well, you know, you went up really high and then you went down. And you did a couple turns, and you went up again, and you went down, and there, meanwhile you're just boring them to death. Like, okay, so why is this such an exciting thing? And I like to think about thermocyclers in that fashion because realistically, you're watching them, and thermocyclers are just probably on the outside. There's probably the most boring piece of equipment that you could ever mess with. But they are amazing in the fact of the stuff that they do, and how they do it, how quick they do it. And so it's just like this roller coaster ride of DNA. All right, so we're going to talk about thermocyclers here, and you'll probably never hear that analogy applied to thermocyclers in your lifetime. So this is the first one. You probably write it down. All right, so now the objectives for us in our talk today, I want to kind of make it brief, is one, how to operate a thermocycler. Thermocycler is kind of a pretty intimidating piece of equipment. It's expensive, incredibly expensive, and it weighs about half a ton, and it's got all these buttons on it. So how do you really operate a thermocycler? And then one of the things that you need to know before you actually run a thermocycler is what settings you should set it at. And one of the settings you should set it at, and you have to find, is the annealing temperature. All right, so the point at which those primers are going to bind, right? So the annealing temperature, also known as TM, I don't know why, maybe melting temperature, annealing, I don't know, and that's going to be out of the primers. So that way, for whatever primers you use, no matter what primers you use in the field, you are able to set the PCR for the correct setting. Let's go back to an earlier time. We were talking about stuff earlier in the section. It's so nice, and we're talking about PCR, and that's the polymerase chain reaction. We said that, you know, this polymerase chain reaction is just a series of heating and cooling steps. We didn't get into any fine details as to what was going on in the PCR reaction. Well, PCR is polymerase chain reaction, so that'd be kind of silly to say polymerase chain reaction. But in this polymerase chain reaction, and we were talking about the steps that were included in that and how you needed certain components to run this PCR. And in case you missed that, you're going to want to go back to the last video talking about master mixes. And it talks about everything that you need to run a PCR reaction. So if you're thinking about running a PCR, here I went again. If you're thinking about running PCR, make sure that you have all the components. And to do that, you want to look at the master mix video. PCR master mix components, we talked about a few of those, what they do, and so you're going to need to know the functions of all those, and yeah, so let's go on. So what does a master mix contain? This is one of the things that we talked about last time. We said that each PCR master mix is going to contain several things, and one of those being a temperature tolerant DNA polymerase. That was either TAC or PFU. And these two things, temperature tolerant, you can throw them in a vat of boiling water and they are perfectly fine. I mean, what else can you put in a vat of boiling water and still work afterwards? Definitely not my computer, but anything you put in a vat of boiling water is going to have to be temperature tolerant and probably water tolerant. And, well, why don't you know, TAC and PFU are both of those. All right, so the next thing you have to add is some DNTPs, essentially just nucleotides with a couple extra phosphates, and these are the building blocks that are going to be used to build your DNA strand. some forward and reverse primers to kickstart the reaction, allow for that DNA polymerase to bind. Some buffer to keep that Taq or PFU polymerase happy. And a PCR cofactor, and that is, in this case, magnesium chloride. Any type of magnesium salt will be fine. Anything with the divalent, actually anything divalent will be fine. Divalent ion magnesium is probably the cheapest one you'll find. So, what are we talking about today? Now, we talked about all that stuff that's needed to run a PCR, and then once we have all that stuff in this tube, we got to do something with it. So we need to figure out how to operate this really bizarre piece of equipment we call a thermocycler. All right, thermocycler, it's also called a thermal cycler. It depends on the text that you're reading it from. Essentially, it's the same thing. They also call it DNA amplifier. And it's got several different names. Just one simple piece of equipment that does a lot of heating and cooling really quickly. So what are thermocyclers? How do you operate one? And how in the heck do you find the annealing temperature? All right. Let me give you a backstory about how we used to do it back in the day. And this was really frustrating. We would have to have 3 individual hot water baths. The first one you'd have to set to that 95 degrees centigrade, and that's that near boiling temperature to get the DNA to separate out. And then you had to have the second one that was set to about 50-60 degrees Celsius or centigrade, and then finally, it depends on ultimately what the primers are that you're using, how long they are, how GC rich they are, all that stuff. And then finally the last one, the third set, has to be set to 72 degrees Celsius. And oh my gosh. All right, so you would have to stand there by this hot water bath, and you'd have to have someone with a timer, and they would set the timer for 2 minutes for the first hot water bath. And once the timer goes off, you switch from the first hot water bath to the second hot water bath. And it has to be quick, right? So you're just like boom, boom. You can't sit there and wait for it. You got to be right there on time whenever it goes off. And then, so then you're there for about 30 seconds. Okay, and set it for 30 seconds. By the time that person sets it for 30 seconds and gets it going, it's probably time for you to go ahead and switch it out to the next hot water bath, right? So you switch out to the next hot water bath, the third one, at 72 degrees Celsius, and then it's got to wait for 6 minutes. And so you're sitting there waiting forever, it seems like, for you to complete one cycle of this PCR step. And not to mention, you have to do this about anywhere between 30 to 50 times if you're using the old equipment, because sometimes those reactions don't make copies if you don't time them exactly right. So you have to do it anywhere between 30 to 50 times, and oh, that will take you all day long. And if you're in the lab for 3 hours, that's pretty much all you're doing when you're doing a PCR step. Let me tell you how nice we have it now. Okay, so now we have a thermocycler. All right, a thermocycler. And this is a piece of equipment that cycles out temperature. Thermo, temperature, cycler, cycle. All right, so this is going to cycle temperatures, going from hot to cold to hot, and it's going to do it really rapidly, just as if you had taken it out of a hot water bath on one side and put it into another one. All right, but the cool thing about it is not that it just changes the temperature really quick, but it knows, like this piece of equipment just knows, like you need to switch it out at what particular time. It's already pre-programmed. You just have to put it in there and hit start. It's just like the most amazing invention ever. And on the outside of it, unless you had to really go back and do that individual hot water bath step over and over and over again, you really don't quite appreciate the laborious task of just switching those test tubes over and over and over again. So this is a tremendous time saver, and you don't have to worry about messing it up. It does it all for you. Glorious piece of equipment. All right, so that is essentially what a thermocycler is. All right, so every lab should have one. All right. So let's talk about what you need to do a PCR reaction. When we talked about what PCR reaction is and the steps that are involved, but what physically do you have to do? What steps do you have to write down in your lab notebook to be able to do a PCR reaction? Well, the first thing you have to do is make a master mix. If you don't already have one, you could buy them offline. They're not too expensive, and if you want to really be cheap with it, you can make your own. And we talked about the components that were needed to make a PCR master mix in the last video, and so if you're curious as to what those components are and what they do, you would want to look back at that one. But essentially, you're going to just take your primers, your specific primers, that is going to be specific for your specific target DNA sequence, and you're going to include those specific primers along with your DNA template, and that is the DNA that you want to be copied, right? So we're going to take those primers, your DNA template, and then we're going to put that in with our master mix, right? Our concoction, our cocktail of PCR. Okay. So we place it all in one of these tubes, and look at this tube here. Just take a look at it. All right. So this tube that you see right here is not your normal microcentrifuge tube. You thought a microcentrifuge tube was small, right? So a microcentrifuge tube is going to hold somewhere around 3 milliliters of volume, okay, for a regular microcentrifuge tube. This one is smaller than that. This one only holds about 0.2 milliliters. All right, so 0.2, that's not even a milliliter. That's not even half a milliliter. Shoot, that's not even a quarter of a milliliter. All right, so that is a really small amount, and it's essentially 200 microliters. Right, so that's the most you can put as far as volume-wise inside your sample. All right, so 2.2 milliliters of this stuff, and these tubes are really small. So we're going to show you some of that when I get to the lab, showing you what these PCR tubes look like, and you have special PCR tube racks and all the other fancy schmancy stuff. All right, so 0.2 milliliters are these PCR tubes. The next thing what you want to do, once you have your concoction in your PCR tube, is when it's all inside there, you're going to want to agitate it a little bit. You want to mix it up so that way all the components are inside the tube and they're evenly distributed throughout the tube. To do that, we're going to use a really fancy technique called the finger flick, okay? The finger flick vortex. All right, so this is just basically you hold the tube with one side, the tip of it with one hand, your lesser of your dominant hand, and then you're going to flick the tube with your pointer finger on your dominant hand. All right, and that is going to agitate what is on the bottom of your test tube, and it's going to mix what's inside of it. They call it the finger flick vortex. All right, more like chaos than a vortex, if you ask me, but that's what it is. Right, you're just mixing up the stuff inside the tube. Next step, after you've finger flicked vortexed your PCR tube, is you're going to collect the entire sample at the bottom of the tube by centrifuging the tube. Centrifuging the tube. All right, so what you see right here to the right is a centrifuge. This is a centrifuge, and specifically it's a microcentrifuge. It holds microcentrifuge tubes. All right, centrifuge. And you will have to know everything about centrifuge equipment, how to operate it, what centrifugal force is, and how that correlates with, all of that stuff. But it's easy stuff. And there's a video on that I've included, so you can watch all the stuff about centrifuging your samples. But essentially, you want to centrifuge your samples, and what that's going to do is it's going to bring back down all of your liquid that may have been like sticking to the side of the PCR tube. whenever you finger flick that tube, so that way it's down at the bottom, so that way you can actually get a micro pipette down in there and get that sample out without having to suck up any air. That's always frustrating. Okay, so you collect it at the bottom by centrifuging the tube. Then we're going to place it inside our thermocycler. All right, so this thermocycler, the ones that we have, are have this magnetic lid, and the other ones need like oil and stuff like that to kind of keep an even temperature across the PCR tubes. This one is pretty fancy schmancy, and you don't have to have any of those. You just put the PCR tubes inside, close the lid, hit the button, and you're good to go. All right, so this is going to make lots of copies or amplify your DNA, and you're going to get lots and lots of copies of that target DNA sequence, just that piece of DNA that's in between those primers, and we call that piece of DNA that's in between those primers the amplicon. Sounds like something off of Transformers, all right? Amplicon. Amplicon. All right, so the amplicon. This is that piece of DNA that is going to be copied. That's a target sequence. Just the target sequence itself. That eventually will be copied through PCR. So it's just that target DNA sequence. And this amplicon, you're going to make tons of it, and over 1 billion copies through this PCR process. So we make all these copies of PCR inside the thermocycler, and it's just going to heat cool over and over and over again. It's real important that you have the settings correct inside the thermocycler whenever you go to run it. So the first thing you want to make sure is that the denature step of it, it will say denature. The denature is going to be set at 95. Most of them are already set to that because, hey, guess what? If you want your DNA to melt, you got to ramp it up to about boiling temperature. All right, so 95 degrees Celsius is probably what it's going to be set to automatically. But if not, you're going to want to go ahead and set it to around 95. Then you're going to have to set the annealing temperature, and that I'm going to show you how to do in a later step in this same video. So you're going to set the annealing temperature on your PCR machine, and that's going to be completely dependent upon what primer sequence that you use. And finally, the last thing is the extension cycle, the last phase where your DNA is actually copied. That's going to be set at a temperature around 72 degrees centigrade. Okay, so 95, whatever your primer is, 72. Okay, so you have those three settings, and you want it to run for about 30 cycles, and that'll give you enough DNA to do whatever you want with. Okay. Now, after you've placed it inside there and it's done its copying, usually what will happen is the thermocycler is like so awesome because after it's done, it's going to like cool that temperature down to like, I don't know, freezing. Okay, so your PCR tubes, when you get them out, if they've been sitting inside the thermocycler, they're going to be pretty cold because it will decrease the temperature as well as increase the temperature. And so you get them out of that heat block, and it's going to be really, really cold, frozen, if not. And that's what you want them to be at. You want them to be ice cold to store your DNA sequence. And so the last step is usually to freeze. But after you get them out of the thermocycler, and if you are going to keep them for a long period of time before sending them off to be sequenced or to inject into an agarose gel, you want to put them in the freezer or in an ice box. Long term, definitely freezer. All right. So you want those in the freezer until you can send those off to be sequenced. And oftentimes, whenever you sequence them, send them off to be sequenced, you put them in a styrofoam insulated box with ice packs and things like that, so that way it stays on ice pretty much the whole time while it's being shipped. So you then send it off to be sequenced. And if you want it to be sequenced, we'll talk about sequencing in a later video. So I'm going to recommend that you watch the instructional video for PCR. This is the Thermocycler or Advotech's instructional video to PCR, and I'm going to have that located on the same instruction sheet as this video, so you can go back there and take a look at that. Okay, so the next thing we need to accomplish, we talked about what it takes to run a PCR or run a thermocycler, and we talked about what a thermocycler is. Now we need to talk about how do you find this annealing temperature. Well, let's go back to what your DNA strand looks like here. So you notice that, you know, we have our one side of our DNA strand, and then we have the other side of the DNA strand, and I want you to look at a couple of things. Well, really just one thing. All right, take a look. Do you remember what these are called? These are the bonds that are located in between bases that hold that double helix together. You remember what that's called? That is called hydrogen bonds. Hydrogen bonds. All right, so these hydrogen bonds are what holds your DNA together, and with AT pairs, you notice that you only have 2 hydrogen bonds, right? Only have 2. How many do you have with CGs? Well, let's look. In CGs, you have 1, 2, 3. So you have basically 33% more hydrogen bond than you did in ATs, right? So when you're pulling these things apart, you have to think about how many bonds am I going to separate, right? And so essentially that's what it comes down to. You know, when you're talking about separating out 2 strands and putting them together, you need to know how many bonds you're dealing with, really. How many hydrogen bonds you're dealing with. And a couple of things that make up hydrogen bonds, and the number of hydrogen bonds, is one, how long your sequence is, right? So no matter what, if you have a long sequence, you're going to have a lot of hydrogen bonds. So the length of your DNA sequence, or your primer sequence, I should say. And finally, how GC rich your primer is. So if you have a lot of GCs, it's going to take a lot of temperature to break those hydrogen bonds, you know. So you're going to want to think about ranking that temperature up a little bit, ramping the temperature up a little bit. I don't know what ranking. I'll ramp the temperature up a little bit to get those hydrogen bonds to split. So how do we find out what to set our temperature to? Well, there's this handy little formula that we're going to use. There's actually 2 of them, but we're only going to concentrate on this first one for problems for testing purposes. All right, so let's say you have a primers or set of primers that are together collectively less than 14 nucleotides in length. Very rarely will you have such an oddity, but just so I can show you how you would find a annealing temperature for something like that, there is a calculation for it, and it's quite simple. It's originally just 2 times the AT sequence, or the number of A's and T's in your sequence, in your primer sequence. You're just going to count up your numbers of A's and T's, and then multiply it by 2. And then you're going to take that number, and you're going to add it to 4 times the number of G's and C's in your sequence. Okay? So however many guanines and cytosines you have in your DNA primers, you'll add those up, multiply that by 4, and then collectively add that to your first number, all right? And that's going to give you your annealing temperature. That's all that's to it. It's just addition and multiplication. That's all it is. Now, if you care to know for everything else what the formula looks like, it is something like this, where you have 65 plus 41 times the quantity of G plus C minus 16.5, and then you're going to divide that by the total number of A's, T's, C's, and G's that are inside your sample, and that will give you your annealing temperature. Still not a bad calculation, but who really wants to do that kind of calculation, right? So there's other things that we can use for that, and let me show you. Well, I'll show you later. I guess we're going to work out a problem first. All right, change of plans. All right, so we're going to work out a practice problem, and we're going to try to see if we can solve the annealing temperature for this primer set sequence. All right, so what I'm going to do is I'm going to use two different colors, and for my A's and T's, I'm going to use blue. All right, so A's and T's are going to be in blue, and my C's and G's are going to be in red. Okay, just so I can keep my numbers straight here. All right, so first thing I'm going to do is I'm going to count up the number of A's and T's I have. All right, so I have one A here, I have a T here, I have an A there, I have a T here, I have an A here, I have a T here, right? So tally up all that. How many do I have? I have 1, 2, 3, 4, 5, 6. I have 6 altogether. So remember the formula. The formula is just going to be, well, I have 12 nucleotides altogether, so that's the first thing. All right, so remember, if it's anything less than 14, I can use this. If it's more than 14, or if it's 14 or greater, I have to use the other formula. All right, but we're going to use this one here. All right, so I have total number of nucleotides is 12, and the formula that we're going to use is this, right? So the 2 times the number of A's and T's I have plus 4 times the numbers of G's and C's I have, and that's going to give me my temperature, my annealing temperature in Celsius, right? So I've got 6 here, right? So I have 6 A's and T's, so that's going to be 2 times 6. All right, and that's going to be added to 4 times the quantity of G's and C's. So we're going to look and we're going to see how many G's and C's we have here. So I have 1, 2, 3, 4, 5, 6. I have 6 also of G's and C's, right? So I would have 4 times 6, right? And that is going to equal my TM, right? So let's find out what this is. So 2 times 6, that is going to be 12. As far as my math goes, that's right. And 4 times 6, I still think that is 24. So I am going to believe it's 24. I'm going to write 24 right here. Okay. And then that is going to give me an answer if I do some simple addition, right? So 12 plus 24, well, that is going to be, what, 36? If my math is right, 36. Okay, so 12 plus 24 is 36. Let's see if I got this right. So 2 times 6 plus 4 times 6. Yeah, so far so good. That's 12 plus 24. Yeah. So my annealing temperature is 36 degrees centigrade. All right. So 36 degrees is what I'm going to set my PCR thermocycler to for the annealing temperature for these primer sequences. Okay. Good. Now, if you don't want to go through the work of, if say you have a 30 base primer sequence, and you have 2 30 base primer sequences, and you don't want to go through the math, do the math, they have this website which is conveniently located both on the PowerPoint and on the study sources section of your information, your sources information, and that is this NebTM calculator. And so this is just a calculator. Just plug in what your primer sequences are. You type in your forward primer, then you type in your reverse primer, and boom, it gives you your calculated annealing temperature for your PCR reaction. There we go again. Make sure, if you don't do it that way, if you don't go through and you run this TM calculator, that you go back and you do it the old-fashioned way with the calculation. Otherwise, you may not get enough of your sample, or you might inadvertently not anneal back that DNA primer, and you might not get anything. And so, you know, it's something that you have to consider, right? It has to be right. That part has to be right. All right, so let's review the things that we learned today. All right, so first off, we learned that a thermocycler is a really awesome piece of equipment, but it looks really boring on the outside, and it automates basically some, or kind of robotically moves some of these repeated cycles of heating and cooling. All right, so heating and cooling. And the typical settings for a thermocycler are going to include, well, your first step is denaturation, and that denaturation step is going to be around 95 degrees Celsius. Then the next step, the annealing, we've learned how to calculate that TM, and so that's going to depend on your primers that you use, obviously, how big that primer sequence is, and how GC rich it is. And then the last step is the extension. The extension is always going to be somewhere around 72 degrees. That's optimal for TAC and PFU polymerase. And we said before, if you're going to do that TM calculation, that is 2 times the numbers of A and T that you have in your primers, plus 4 times the number of G's and C's that you have in your primers. And that's only used if you have less than 14 nucleotides. Anything more than 14 nucleotides, you gotta go and do the other calculation, or you go to NEB's website and you plug in the sequence yourself. Okay, so some things to take home with you. The first thing is, what is a thermocycler? You need to know what a thermocycler is, right? You need to know how to calculate the easy method for the less than 14 sample, or less than 14 nucleotides, right? So you need to know that. And then you'll want to be able to organize in your head the steps that's required to perform a PCR reaction, the one that we went over earlier. And that video is located on the same sheet here, and so you can get on there and you can watch those steps over and over and over again, or watch this one over and over again, whichever one you want. The other one's probably faster, but watch those steps. Make sure you know what you're going to be doing at each step. That's always important whenever you are doing a lab. All right, so make sure you're consistent with that. All right, so I've taken enough of my time and your time, so I am going to head out. And if you have any questions, please feel free to let me know. And you can always go back, rewatch the video if you so desire. All right, thank you so much.